Hello, my dear students. Welcome to my class. Last day, we have discussed about the machinery of the enzyme. That the enzyme prepared for the DNA replication. And now, today we are going to discuss with the process of DNA replication. So today we are going to take process of DNA replication. So ये जो प्रोसेस ऑफ डीएनए रेप्लिकेशन है, बोर्ड पॉइंट व्यू ऑफ एग्जामिनेशन के लिए ये कभी-कभी 5 मार्क्स का क्वेश्चन भी पूछा जाता है. So in this lecture we will see that how can we make the questions from this topic और हमें ज़्यादा कहाँ पर कंसंट्रेट करने की ज़रूरत है. ठीक है बेटा? हमारे पास जो डीएनए की रेप्लिकेशन है, सपोज 2 मिनट के लिए हमारे पास एक डीएनए मॉलिक्यूल है. सपोज दिस इज अ डीएनए मॉलिक्यूल, linear DNA molecule, or we can say it can be circular DNA molecule. This DNA molecule has 2 strands. एक strand 5 days की 3 days में लिखा है, दूसरा इसका अपोजिट होता है, ये 3 days to 5 days. In this what we are having the hydrogen bases which are joined by hydrogen bonds like these are hydrogen bonds. Suppose these 2 strands are just bind to each other to form a DNA molecule. In DNA molecule, the point where from the replication actually begins, that point is known as ORI site. So we can say that DNA replication start point. So here in this DNA molecule, there should be some point where from the DNA replication starts. That point is known as actually the ORI site. So the point at which DNA replication starts, that point is known as ORI site, or we can say origin of replication, or origin of replication. As we know, this DNA molecule is not a short molecule or little molecule. ये बहुत बड़ा molecule होता है, इसलिए इसमें ORI site एक ही नहीं होती है, बहुत सारी होती है. लेकिन एक segment which having only one ORI site, that is called as replicon. Let's write it here. So a DNA segment having only one ORI site, it is known as replicon. आपसे MCQ में ये पूछा जाता है, what is replicon? Replicon is the segment of DNA actually in which only one ORI site is there. Suppose here this point is the site where there is this ORI site. So at this site, this is called as ORI site. At this site, the replication starts actually. Is this clear? From this ORI site, what we need to concentrate here, it is that the enzyme, which we have already discussed, that enzyme will act on it. One enzyme, that is called as helicase. It is helicase enzyme, or we can say it's unbinding enzyme. ये जाके वहाँ ORI site पे gets bind and it breaks the hydrogen bonds. Let me show you how. So in the third step, helicase acts on ORI site and uncoils the 2 strands. The 2 strands by breaking hydrogen bonds. Is this clear? So after this structure will be like that. When the hydrogen bonds are getting broken, this structure will It will be like that. So this is the origin of replication. These are hydrogen bonds. This is the origin of replication. These are hydrogen bonds. But this segment in which these hydrogen bonds are broken down, this is actually the unwinding of the two secrets. So here is one origin of replication. Here is another origin of replication. So this point, where from the replication starts, this is called the actually origin of replication. There is O on my side. Suppose this is the 5-day end, and obviously this will be the 3-day end. Obviously we can see this is 3-day end, this will be 5-day end. Is this clear? Now coming to only the half part of this DNA molecule, we replicate. We need to understand the replication. So by this we can see this molecule like this. This is a molecule, and this is one replicon. In this replicon, we see these are the hydrogen bonds, and from here this point, the hydrogen bonds are not there. These two strands has been uncoiled. These two uncoiled strands, this is 5-day end, this will be 3-day end, this will be 3-day end, this will be 5-day end. Must remember the orientation or polarity of the two strands. Now what is happening that this single strand, these are the two strands without hydrogen bond. These are just their tension of coiling is removed by tension of coiling is removed by an enzyme also we have studied by an enzyme that is called as topoisomerase. That is called as topoisomerase. So जो अगर हम इस किसी रोप में जो दो थ्रेड्स होते हैं उनको ऐसे हम खींचे फिर छोड़ दें तो फिर से कॉइल हो जाएगा. इसी तरह से डीएनए भी कॉइल होते हैं. इसको ऐसे खींचे, हाइड्रोजन बॉन्ड्स बिल्कुल अलग हो गए, फिर से ये वापस आएगी और कॉइल करेगी. लेकिन उसको बचाने के लिए एक एंजाइम वहां पर होता है दैट इज कॉल्ड एज द टोपोआइसोमेरेज एंजाइम. दैट इज एंजाइम टोपोआइसोमेरेज. दिस जस्ट रिलीज दिस टेंशन ऑफ द कॉइल बाय व्हाट? वो इनको थोड़ा कट करके फिर से जोड़ देता है, कट करता है फिर से जोड़ देता है ताकि जो रिकॉइलिंग का टेंशन है वो कम हो जाए. समझ में बात आई? आफ्टर दिस टू स्ट्रेंड्स दे आर अनस्टेबल. कॉइलिंग को टेंशन छोड़ दिया लेकिन फिर से हाइड्रोजन बॉन्ड बनाएगा, क्या होगा? उसको फिर से हाइड्रोजन बॉन्ड ना बनाने के लिए इसमें एक प्रोटीन लगता है दैट इज कॉल्ड एज सिंगल स्ट्रैंड स्टेबलाइजिंग प्रोटीन. ये एक ऐसा प्रोटीन है जो इस सिंगल स्ट्रैंड को स्टेबलाइज करता है ताकि फिर से हाइड्रोजन बॉन्ड अपने दूसरे की साथी के साथ ना बनाए. So these are the two enzyme and this is the second protein. What are the functions of topoisomerase in order to release the tension of coiling? And what is the function of single strand stabilizing protein? It stabilizes the single strand. It stabilizes the single strands. किस चीज से? ताकि वो फिर से हाइड्रोजन बॉन्ड ना बनाए. ठीक, तो अब हमारा यहां तक ये स्ट्रक्चर बन गया. ये ऑरिजिन ऑफ रेप्लिकेशन है. बट ये जो स्ट्रक्चर बना, दिस स्ट्रक्चर that after this what we call as unwinding of these two helices, this structure is known as the replication form. So this structure is in Y-shaped form. It's a Y-shaped structure. Y-shaped structure formed after unwinding of two helix of two strands is called replication form. It is known as replication form. As I have catalyzed by a main enzyme that is called as DNA polymerase. This DNA polymerase is actually DNA dependent DNA polymerase because यहाँ पर जो DNA खुद को नहीं बनाता है, it is called as autocatalytic role in which DNA itself replicates itself. So that means the DNA polymerization, it is catalyzed by the enzyme that is called as DNA polymerase. It is actually DNA dependent DNA polymerase. It is DNA dependent DNA polymerase. And always polymerization takes in a particular direction, that is 5' to 3' direction. Always polymerization takes place in 5' to 3' direction only. So you have to remember. So if we see this spread, let me rub this board first. You just take a screenshot of the board. आप अपना स्क्रीनशॉट ले लीजिए. तो मैं बोर्ड को रब करके इसको थोड़ा सा आगे ले जाता हूँ. अब हमारे पास जो स्ट्रक्चर है वो है रेप्लिकेशन फॉर्म. रेप्लिकेशन फॉर्म में क्या होता है, यहाँ पर डीएनए की पॉलीमराइजेशन होती है. तो आपको थोड़ा सा डायग्राम में भी याद रखना पड़ेगा कि क्या क्या चीजें होती है. हाँ, क्लास में होते तो बहुत मजा आता, लेकिन क्लास में नहीं हो ना, अभी मजे कर रहे हो. ठीक है, तो मैं क्या काम करता हूँ बेटा, यहाँ पर ये रेप्लिकेशन फॉर्म बना देता हूँ. तो दिस इज द रेप्लिकेशन फॉर्म. ये इसका वाई शेप स्ट्रक्चर है. दिस इज आई थिंक इट वाज 3 days. सपोज दिस इज 5 days and this will be 5 days and this will be just 3 days. So this is the replication form. These are the hydrogen bonds. It has not been unwinded yet. And this is the origin of replication here. Now DNA polymerization will take place. The DNA polymerase will act here, and polymerization will take place. For polymerization, for DNA polymerization, we need energy because it is energy consuming process and substrates. Substrates कौन सा है हमारे पास? अगर substrates हम बोले तो substrates हमारे पास है DNA पॉलीन्यूक्लियोटाइड्स. यानी न्यूक्लियोटाइड्स हमें चाहिए ताकि हम DNA का चेन बनाएं. तो इन substrates के लिए हमें चाहिए, एक तो चाहिए डीऑक्सी एडिनाइन adenosine, sorry, it is deoxyadenosine triphosphate. यह हम ऐसा नहीं लिख सकते हैं. जनरली जो सबस्ट्रेट्स होते हैं, these are actually deoxynucleoside triphosphates. Generally we can say these are deoxynucleoside triphosphates. So these are deoxynucleoside triphosphates. That is the first one is deoxyadenosine triphosphate. We can see this is D-adenosine triphosphate like this. Triphosphate means these are having 3 phosphate groups here. But to bind into the this DNA molecule, 2 phosphate groups will be released and they provide energy for the process. So here DNA polymerization we need energy. Energy कहाँ से आती है, वो सबस्ट्रेट कहाँ से आता है? हमारा सबस्ट्रेट deoxyadenosine triphosphate है. एक तो ये सबस्ट्रेट भी काम करता है and energy भी प्रोवाइड करता है. So these deoxynucleoside triphosphates they provide energy. They provide energy as well as substrates for polymerization. It can be written as DGTP. Number third is deoxycycline triphosphate. It is DCTP, and also you can have deoxythymidine triphosphate. Triphosphate, and it can be written DTTP. So these substrates, they can function as both as substrate as well as energy, because polymerization, this DNA polymerization is energetic unique process, and for that energy and substrates, these deoxynucleoside triphosphate, they can provide energy as well as substrate for polymerization. This is the most important point you have to remember here, क्योंकि जब DNA polymerization होती है तो पॉलीमराइजेशन के लिए आपको सबस्ट्रेट चाहिए होगा, एनर्जी भी चाहिए डीएनए मॉलिक्यूल बनाने के लिए। तो ये जो हमारे पास सबस्ट्रेट्स हैं, ये बोथ एक्ट करते हैं, सबस्ट्रेट भी और एनर्जी भी। डीएनए पॉलीमराइजेशन तब तक स्टार्ट नहीं हो सकती जब तक ना हमारे पास एक आरएनए का छोटा न्यूक्लियोटाइड सीक्वेंस बनना चाहिए, उसको हम बोलते हैं आरएनए प्राइमर। तो इसके बाद में होता है हमारे पास यहाँ पर फॉर्मेशन ऑफ, इसके बाद में फॉर्मेशन और आरएनए प्राइमर। ये आरएनए प्राइमर क्या है? इट इज एक्चुअली द स्टार्टर ऑफ द रेप्लिकेशन। डीएनए रेप्लिकेशन ऐसी ही स्टार्ट नहीं होती है जब तक ना पहले ये आरएनए प्राइमर बन जाए। अब ये आरएनए प्राइमर क्या है हमारे पास, मैं बताता हूं आपको। एक सेकंड, ये जो हमारे पास आरएनए प्राइमर है, it is 6 to 7 nucleotide base sequence which start the process of replication. It is 6 to 7 base sequences of nucleotides which starts the process of replication. Process of replication. और ये जो आरएनए प्राइमर बनता है, it is because of the enzyme that is called as primase. So primase is one enzyme which catalyzes the formation of RNA. It catalyzes the formation of RNA primer. So this is a short sequence of these 6 to 7 base sequence of nucleosides taken. Draw this, suppose this is the RNA primer. This RNA primer is a short sequence of these 6 to 7 base nucleosides. It also is having thioate end at 3 days. This is the thioate end, this will be the 3 days end. At 3 days end, OH is free, and at 5 days end, there is a phosphate group which is not free. So we know that all these new nucleotides are added onto the 3 days end, or we can say onto the 3 days carbon atom of the first nucleotide. So these are the nucleotides actually. So new nucleotides will be added at this position only, and the polymerization will be in the direction of 5 days to 3 days direction. So let us place this RNA primer into this. So if this is the RNA primer, it will be placed opposite to the end. Suppose this is the thioate end. When we draw this, what we call it primer, it will be 3 days end here, thioate end here. But nucleotide will be added here. So we don't need the nucleotide to be added here. So this primer will not be placed here. It will be placed here. So it will be placed like this. So this will be thioate end. This will be just 3 days end. This is the OH group. This is the 3 days end. So primer is just place it here. On to the next strand, there will be the again binding of the primer here. So here the primer will be binding here, because on the opposite free element there will be third element, and it will be the direction of this. This will be three element. So always the direction of the polymerization will be 5 days to 3 days. 5 days to 3 days. So here also the direction of the polymerization is from 5 days to 3 days direction. So these are the primers. So this is the binding of the primer onto this strand. In this, what we see here on the strand, there is 5 days to 3 days, and this is 3 days to 5 days. The new nucleotides will be added here on the 3 days. Suppose this is one of the nucleotide, this is second of the nucleotide, this is third of the nucleotide, this is fourth of the nucleotide. From here on, this strand will complete. Is this clear? So this strand will be completed here, but this strand will add a nucleotide towards the inside. So many of the nucleotides will be added here. So it will reach the ORI side where there is unwinding has not been taken place yet. So this is the first phase of the replication. Now we'll see that these hydrogen bonds are further unwind. Then this two helix or two strand, they will open up again to some distance. So in this we'll see this strand will be like this. So these two strands open further. As we know that DNA replication in that DNA molecule is bidirectional, and in this DNA replication all the DNA molecule gets replicated. All the DNA, all whole DNA molecule will be replicated. So here the strands are open further, then it will be like that. So we have made this replication here. Suppose this is the 5 day event, this will be 3 day event, this will be 5 day event, this will be the 3 day event. Already we have made. Suppose here यहाँ तक already replication हो चुकी है. This part has been opened further. तो यहाँ पर मैं क्या काम करता हूँ? यहाँ पर मैं ये primer लगा देता हूँ blue color से. This is 5 day event and this will be 3 day event. And here also here was the primer. In this primer, this is 5 day event, this will be 3 day event, and the nucleotides already has been added here. So this is one nucleotide, second nucleotide, third nucleotide, fourth nucleotide, like that only. Here is another nucleotide, one nucleotide, one nucleotide, one nucleotide till here. Now this is the portion we can open up further. Now in this strand, which is 5 days to 3 days strand, the polymerization will be continuous. It will be continuous. It will be continuous. But into the opposite strand, there will be then again the format of primer, because Discontinuous polymerization is 3 days to 5 days direction of the template strand. In this discontinuous polymerization, that is 3 days to 5 days template strand, there is a formation of the fragments of DNA. There is formation of fragments of DNA. These fragments are known as Okazaki fragments. These are known as Okazaki fragments. So these Okazaki fragments, these were discovered by Okazaki actually. So these are actually the fragments of the DNA which is formed in the 3 days to 5 days template strand. So that is why this discontinuous polymerization or discontinuous strand, this is known as lagging strand. This is known as lagging strand. You will be asked in the examination, in which strand there is discontinuous polymerization and in which strand there is continuous polymerization. After that, when the whole DNA molecule gets replicated, you just take the screenshot. Take the screenshot. Okay. When the whole DNA molecule is getting replicated, the whole DNA molecule gets replicated in the both direction. So DNA replication is bidirectional. So after that, the whole DNA molecule gets replicated. The whole DNA molecule is getting replicated in both directions. That is DNA replication is bidirectional. That is DNA replication is bidirectional, and always DNA replication, it polymerizes the DNA molecule in the direction of 5 days to 3 days direction. So it will be 5 days to 3 days direction. So when the whole DNA molecule gets replicated, it gets replicated in both directions. That is DNA is bidirectional, and always this DNA which is got replicated, sometimes some wrong bases will be entered in these positions. Some wrong bases are getting entered into these positions. They are just getting removed by some enzyme, and the particular base and the right base is introduced in this. That is called as DNA repair. So if a wrong base is introduced, if a wrong base is introduced in the new strand, sometimes the wrong base gets introduced into the new strand. Then what has happened? Then the DNA polymerase first. Then the DNA polymerase first can do proofreading. What is proofreading actually? It can remove the wrongly added base and replace it with the right type of base. That is called as DNA proofreading. So DNA proofreading is just can be catalyzed by this DNA polymerase 1. Next thing is that this DNA molecule gets replicated. In this DNA molecule, we get this leading strand and lagging strand. After that, these primers are removed. fragments are joined by DNA ligase enzymes. DNA ligase enzyme, and a new molecule of DNA will be formed. Is this clear or not? So this is the process of the replication for DNA. Now, any failure in the cell division, if there is a failure in the cell division after the replication of DNA, it will lead to the polyploidy. So let me write it there, some most important point here. Any failure in the cell division after replication results into polyploidy. This is the most important. Polyploidy means a cell having many chromosomes. So because DNA replication, the chromosome number is added, but when cell is not dividing, then chromosome number which has been added, it will form the poly. Poly means many, ploidy means a chromosome. It will have many chromosomes. One problem is being asked there, that this DNA replication, it occurs in all organisms. This DNA replication is very accurate, and it is in a particular type a species can replicate its DNA molecule. Let us take an example of the Escherichia coli. Just take a screenshot of this. We'll have some problem here because it is being asked in the exams. So let me discuss the problem that this DNA replication is the enzyme which has been used in this DNA replication. These are very accurate and they polymerize the DNA very accurately and also in a particular time. So let me take an example. Every species is having its own time for its replication and accuracy. So we can take an example of Escherichia coli. Escherichia coli has 4.6 into 10 raised to power 6 base pairs of DNA. So we can measure the DNA by base pairs. If we see how much time it will take for the replication, we know that the Escherichia coli can complete its replication in 18 minutes. So Escherichia coli can complete its replication in 18 minutes. Let us change these 18 minutes into seconds. It will be 18 into 60 seconds. Is this clear? Now we will see the rate of polymerization, or we can say the how many in 1 second how many base pairs are formed. It's called as rate of polymerization. That will be rate of polymerization. In rate of polymerization, we can see that it will be equal to total number of base pairs divided by time taken. Here we have both these things. It will be 4.6 into 10 raised to power 6 divided by 18 into 60. It's like that only. Is this clear? Now let me simplify like a math cell. It will be 46 into 10 raised to power 5. It will be 46 into 10 raised to power 5 first. Is this clear? Divided by 18 into 60. Why is 46 into 10 raised to power 5? Because I have just removed this point. If we remove this point, then ये रेट ऑफ पॉलीमराइजेशन। सो कितना, अगर एस्टेरिया कोलन को देखें तो रेट ऑफ पॉलीमराइजेशन इज 2000 बेस पेयर्स पर सेकंड। सो बेटा यहां पर हमारा डीएनए रेप्लिकेशन पूरा सॉल्व हो गया और हमने डीएनए रेप्लिकेशन का प्रोसेस भी पढ़ा और एस्टेरिया कोलन में रेट ऑफ पॉलीमराइजेशन कितना है आप ह्यूमन बीइंग्स का भी रेट ऑफ पॉलीमराइजेशन निकाल सकते हैं। तो आप इसका स्क्रीनशॉट ले लीजिए और आज का लेक्चर यहां पर खत्म करते हैं। तो आज इस लेक्चर के साथ में आपका एग्जाम पोर्शन भी खत्म होता है। टू प्रिपेयर फॉर द बोर्ड एग्जाम, आपका बोर्ड एग्जाम का पोर्शन इधर ही तक है कि मैशनरी ऑफ द एंजाइम और डीएनए रेप्लिकेशन। सो टुडे वी कंप्लीटेड दैट पोर्शन। यू जस्ट कंसंट्रेट ऑन टू योर these board questions and the back of biology you are having that what they called as, इसको क्या बोलते हैं बेटा, जो नोट्स है ना बायोलॉजी के उसमें से आप ले लो। ओके, थैंक यू, बाय, टेक केयर।